Hi everyone, so my name is Isabella Wagner and I'm going to be doing my presentation on how mutations in the CYP21A2 gene lead to adrenal insufficiency. So as a quick overview, I'm going to be focusing on 21-hydroxylase deficiency in this presentation, which is a disorder that affects the adrenal cortex. I didn't do a research project, I'm doing a literature review, so I'm going to be compiling data of different CYP gene mutations and the frequencies at which they occur. I'm really interested in why the mutation frequencies will differ, which specific gene mutations lead to adrenal insufficiency, and analyze the genotype-phenotype correlation. So let's get into some background and introduction. So starting off the 21-hydroxylase deficiency, this is the most common form of congenital adrenal hyperplasia. It affects how the adrenal cortex functions and produces aldosterone and cortisol. The mutations in the functioning of the 21-hydroxylase enzyme is controlled by mutations in the CYP gene. As a quick aside, I'm going to call it the CYP gene just to save some time. And I'm going to get into the pathway in the next slide right here so that it makes more sense. Sorry. So the 21-hydroxylase enzyme is, in a sense, it's an intermediary step in producing aldosterone and cortisol. So as you can see, it converts progesterone to deoxycorticosterone and 17-hydroxyprogesterone into 11-deoxycortisol. So when it is not functioning adequately, the production of aldosterone and cortisol will suffer, and most of the energy in this pathway will be put towards producing androgens to the right, like estradiol and testosterone. So for the salt-wasting form, the three different forms of 21-hydroxylase deficiency. The first one is the classic salt-wasting form, and that is when you have a completely non-functional 21-hydroxylase enzyme, so no aldosterone or cortisol can be produced. Some side effects of this would be hyponatremia, hyperkalemia, and a low blood volume leading to hypovolemic shock, since sodium cannot be reabsorbed, so water will remain with it in the filtrate. The simple viralizing form is very similar to what I just mentioned, except the mutation in the CYP gene will render the 21-hydroxylase enzyme still somewhat functional, maybe only 1% functionality, but there will still be a little bit of aldosterone and cortisol produced. I have a picture of testosterone on the right just to remind myself that in both of the forms I just mentioned, the salt wasting and the simple viralizing, the external genitalia of females will be ambiguous and males will have an enlarged testis, and this is due to excess production of androgens. The non-classic form is the least severe of the three forms. The 21-hydroxylase enzyme functionality is better than what I just mentioned, and a common side effect of this would be hirsutism, which can be visualized as excess hair growth on females. So a little bit about the CYP gene. It belongs to a family involved in cholesterol production and drug breakdown, and it lies within the histocompatibility index of chromosome 6. That's really important when we get to gene conversions, which I'll explain later in the analysis. Some background on mutations. Different mutations will affect the functionality of the enzyme differently, depending on if they're large deletions, just point mutations, maybe missense mutations. And the severity of 21-hydroxylase deficiency is determined by the least severely affected of both genes. So that's how the phenotype will be determined. And 90% of the mutations I'm going to talk about actually result from gene conversion events. This is when you exchange material with a pseudogene. A pseudogene is basically, in this case, a CYP gene with errors and unwanted sequences. And when it exchanges material with a normal CYP gene, then it will basically replace part of the normal CYP gene with a mutated sequence. And the reason that these occur so frequently is because in the histocompatibility index, there's a lot of chi-like sequences that promote homologous recombination. So my goal of this presentation is really just to determine what mutations lead to adrenal insufficiency, why their frequencies differ, and the extent to which phenotype can predict genotype. So let's get into some mutation prevalence and establishing the genotype-phenotype correlation. So I'm going to start out with the family study done by Yadav and al., in which he looked at how children will inherit the CYP gene mutations from the parents. And what he found is that in general, if the parents were affected with mutations, the children will have them as well. But some mutations just had no parental origin, like they were sporadic, you could not predict them. White found the same thing. He found that gene deletions on parental genes are usually linked to the classic form of the disorder and are also sporadic. So parental studies are definitely necessary to determine how the mutant allele was segregated during meiosis, but they won't always accurately predict the phenotype of the children. So I'm going to look at the chart of Yadav and al. A little more just to expand upon what I just stated. So specifically, let's look at families 5 and 6. In family 6, we can see the parents had these mutations, but when we look at the mutations that the children had, one of them has an I172N mutation, which is in neither of the parental genotypes, so thus sporadic, hard to predict. Same case in family 5, we look at the parental genotypes, and then we see that the children had an R356W mutation and a Q31A mutation. So something to consider there. Some mutations are random and can't be predicted. And also, just because the parents had a mutation did not necessarily mean all the siblings would be affected. If we look, we had 5 children, but only 2 affected here. But over here we had 4 children and all 4 affected. So it honestly just varied from family to family in this study. So from this, I concluded that genotype-phenotype correlation is difficult to establish for 21-hydroxylase deficiency. Adding on a little more to this, Milisic and al. Also supported these findings, found that some mutations are sporadic. Something interesting in his study is he found that switching from leucine to proline at position 129 and serine to proline at position 165 were the most deleterious mutations and associated with the salt-wasting form. Kind of unexpected, because you wouldn't think that a single point mutation would cause the enzymatic function of the 21-hydroxylase enzyme to be basically non-existent. And it makes sense that the salt-wasting form would have the highest mutation detection rate because the phenotype is so severe that it's easier to see what caused that than the other types, for example. So the general trend that I found from all these studies was that the severity of 21-hydroxylase deficiency was proportional to the amount of enzymatic activity compromised by the CYP gene mutations. So when I was looking at mutation frequency in all of these studies, I found that there was 3 mutations that kind of jumped out at me. They were present in almost every single study. I even actually bolded in which this particular mutation was the most frequent in the entire study. So I really wanted to just analyze why these mutations were more frequent than others, what types of 21-hydroxylase deficiency that they are associated with, and just expand upon that. So Waddell and al. Sequenced full-length pseudogenes. They found that the intron 2 mutation was the most prevalent and associated with the classic form. And that does make sense because introns are non-coding sequences. You don't want them to be transcribed or translated since the protein product produced from that would almost certainly be non-functional. And in an intron 2 mutation, the second intron is incorrectly spliced, leading it to probably be transcribed and translated. Therefore, the 21-hydroxylase enzyme is non-functional, hence the classic form. So I want to analyze the isoleucine to asparagine mutation at position 122 a little more. This is the only mutation that's solely associated with the simple viralizing form. When you think of simple viralizing form, everyone looks at this mutation. And this is because it produces an enzyme with 1% of normal activity, and I really just wanted to look at why. So in a study done by Amor and al., he found that this mutation is normally present in the pseudogene, and because usually when it undergoes gene conversion, it's transferred along with mutations in exons 3 through 6. This could explain why enzymatic functioning is so compromised, because it's normally transferred along with a series of other mutations that will affect functionality there. But I also wanted to look at the biochemical structure just to understand this a little bit better. So when we look at isoleucine, the side chain of that is very nonpolar, made of hydrocarbons right here. But when we look at the side chain of asparagine, it's made up of a carbonyl group and another nitrogen group, so making it very polar. So when you switch from a nonpolar to a polar amino acid residue, this will definitely change the active site of the enzyme, and probably the substrate will not be able to bind very effectively, or even at all in some cases. So this could also show why enzymatic functioning is so compromised from this mutation. Now let's get to something really interesting. So the proline to leucine mutation at position 30. And this is actually associated with both the simple viralizing and the non-classical form, which seems kind of counterintuitive because you wouldn't think that the same mutation would be associated with 2 different forms. So why does the same amino acid substitution cause such varying functionality of the 21-hydroxylase enzyme? So this is why. In a study done by Araujo et al., he found that this mutation was often present along with substitutions in the pseudogene promoter sequence. And he found that promoter substitutions can actually decrease transcriptional activity up to 5-fold. So that means that this mutation is normally present with promoter sequence substitutions, and this would be why the phenotype is more severe than what the genotype predicts. Because the promoter mutation in combination with the amino acid substitution would generate it would be far more compromising to enzymatic functionality. So that is why it's also associated with the simple viralizing form. When the promoter sequence mutation is non-existent or not as severe, then it could possibly be associated with the non-classical form. So let's analyze mutation frequency a little bit more. In a study done by Tusi, Luna, and White, they found that gene conversions actually occur by a mechanism separate to crossing over, and we don't fully understand what this mechanism is, but just something to consider. And when we look at the promoter sequences of pseudogenes, promoter sequence mutations may produce non-functional pseudogenes, and these pseudogene mutations can be transferred via gene conversion. And obviously, the severity of the promoter sequence mutation will contribute to phenotype. And so, as a general hypothesis, I do believe that the intron 2, the I172N, and the P30L mutations probably have a higher probability of being associated with promoter mutations. And this could be why they all compromise enzymatic activity so much and why they are so much more frequent. Perhaps whenever there's a promoter sequence mutation, it's more likely to be seen in combination with these mutations. So why do unequal crossover events only account for the remaining 10% of CYP gene mutations? And I believe that this could possibly be because the mismatches caused by unequal crossovers are better recognized and repaired by the cell machinery. Perhaps cellular processes involved with crossing over are better regulated and modified than those in gene conversions. So that brings me to our conclusions, and the conclusions that I gathered from all the studies I looked at is that the greater frequency of the intron 2, I172N, and P30L mutations may result from them being more associated with pseudogene-specific promoter sequence mutations. Perhaps when a pseudogene promoter sequence mutation is transferred via gene conversion, it is more likely to be transferred with one of these mutations right here. That just seems to be why their frequency could be so high, especially for the P30L mutation. They actually found that this is indeed true for that, but we need to do future studies on how promoter sequence mutations relate to the intron 2 and the I172N mutation. But point mutations coupled promoter sequence will definitely lead to more severe phenotype than what the genotype suggests, and this will explain why phenotype correlation is difficult to establish. You'll have to look at the whole region of the promoter sequence to really confirm this. And then my last conclusion is that the CYP gene mismatches from unequal crossovers are likely to be better recognized and repaired by cellular machinery than those occurring from gene conversions. So in a real-world application, I think future studies should really focus on sequencing the entire promoter region of affected individuals with 21-hydroxylase deficiency. And I think it would be really good to determine the actual mechanism of gene conversion and how the cell regulates it, just to see if what I concluded was indeed correct. I think the study was really helpful in that it allowed me to understand the genetic processes of the most common form of congenital adrenal hyperplasia, and all the mutations I discussed will cause adrenal insufficiency. So for some acknowledgements, I would like to thank Ms. Jamie Moon of the UNF Bio Department for her help in guiding me to write this paper. Thank you so much for being a great mentor. And here is my bibliography of the studies I used, and that is about it. So I hope you all enjoyed my presentation, and I hope everyone has a great week.